The Hidden Janitor, a story of autophagy and cellular renewal. Once upon a time, within the microscopic kingdom of the cell, there existed an unsung hero, autophagy, a silent guardian tasked with the never-ending job of maintaining order amidst chaos. For years, this process went unnoticed, much like a janitor cleaning silently after everyone had left. But as scientists peered deeper into the workings of cells, they began to understand that autophagy was far more than just a passive cleaner. It was a highly orchestrated, essential survival mechanism, especially in times of cellular stress. Autophagy, from the Greek for self-eating, is not random destruction. It's precise. When unwanted or damaged parts build up, be it a malfunctioning mitochondrion, a worn-out lysosome, or long-lived proteins past their prime, autophagy steps in with purpose. This targeted cleaning process isn't a blunt sweep, but rather a refined operation involving specific forms such as mitophagy, removal of mitochondria, and lysophagy, removal of lysosomes. But how does this happen? The journey begins with the formation of a small cup-shaped membrane called the phagophore. This membrane expands, wrapping itself around the cellular debris like a gift being packaged. Once sealed, it becomes a double-membraned autophagosome ready for delivery. Its destination? The lysosome, the cell's digestive hub. Upon fusion, the contents are broken down and recycled, nutrients are reclaimed, and cellular balance is restored. Unlike other forms of protein degradation such as proteasomal breakdown, autophagy is unique in its ability to engulf entire organelles and bulk cytoplasm. It's essential for survival during starvation, as it recycles internal components to sustain life, and in senescent cells, it ensures turnover of old, potentially toxic, cytosolic materials. Modern research has revealed the intricacies of this process, particularly in mammals. Scientists have uncovered a complex cast of molecular characters working behind the scenes. One of the central directors is the mTOR pathway, more specifically, mTORC1, mechanistic target of rapamycin complex 1. When nutrients abound, mTORC1 is active and suppresses autophagy by phosphorylating critical components like ULK1 and ATG13, preventing the machinery from starting. But when the feast turns to famine, mTORC1 is silenced. This repression frees LK1, which activates itself and triggers the cascade of events needed to form autophagosomes. Still, mTOR's silence is not enough. Another messenger steps in, EF2-alpha, whose phosphorylation signals a halt in protein synthesis, redirecting the cell's energy toward survival rather than growth. During times of energy stress, such as exercise, fasting, or metabolic strain, AMP-activated protein kinase, AMPK, senses the drop in cellular energy. Like a fire alarm in a burning building, AMPK activates and further fuels autophagy initiation, ensuring the cell adapts and survives. And the story doesn't end there. Recent discoveries have revealed a master switch in the nucleus, transcription factor EB, TFEB. Under normal conditions, TFEB is restrained in the cytoplasm by, you guessed it, mTORC1. But during stress, when mTORC1's grip loosens, TFEB journeys into the nucleus and activates a wave of genes responsible for autophagy and lysosome biogenesis. It's the ultimate backup system. When the cell needs to reorganize its internal waste management, TFEB leads the charge. From defending the cell against pathogens to fine-tuning immune responses, from countering kidney damage to clearing metabolic waste, autophagy is at the heart of cellular resilience. In the symphony of life within every cell, autophagy is both the conductor and the cleanup crew, harmonizing survival and renewal with astonishing precision. Though once hidden in the background, it now stands in the spotlight as a cornerstone of cellular health and a potential key to treating diseases once thought untouchable.